Hello everyone, my name is Brent Hill and I am a third-year agricultural and environmental plant science major at Cal Poly San Luis Obispo. Today I will be teaching you how to conduct your very own insect collection. Now before we get started collecting any insects, every good bug collector needs their equipment. Now what I have right here is my handy dandy satchel. Now I have a couple of things ready to go, and that first is a killing jar. A killing jar consists of any type of jar, mason jar, an empty peanut butter jar, anything that you can get your hands on, and a piece of paper towel in there dosed with ethyl alcohol or isopropyl alcohol if you have it available to you. Now the concept behind this is that you will trap the bug inside of the jar and it'll knock them out so that you can then freeze them later. The next thing that we have in our handy dandy satchel is an aspirator. An aspirator is a great tool for collecting small, soft-bodied insects. How an aspirator works is there is a metal end here that you're going to be placing near the insect and a sucking tube that you'll be able to use to suck the insect into the little vial there without getting it in your mouth. This is an incredibly useful tool and I highly recommend getting one for your collection. Now for the smaller bodied insects, we would want to put those in a vial. Now this vial has ethyl alcohol in it. Like I said, you could use isopropyl alcohol if you do not have the ethyl alcohol available to you, but pretty much this is a great tool to have when collecting those smaller insects that you don't want to take up a whole lot of space for. And finally, one of the best tools for catching some really cool looking insect is a net. Now this net is very helpful and useful when getting a long reach on something that you don't think is going to get away. So this is great for butterflies, Lepidoptera, and wasps. This video documentary is really designed for anyone who just wants to get started in the world of entomology. That could mean a college student getting a collection for their entomology class, or perhaps an adult that's wanting to spend time with their children. Anyone who wants to get involved in the field of entomology, this video is for you. Now for the first environment that we'll be collecting in, this is an agricultural field and there's no shortage of insects here. This is the Cal Poly Organic School Farm, and the temperature today is around 70 degrees, slightly windy, and this is in the middle of April, so bugs are flying around everywhere, and it's absolutely going to be perfect for our collection. Alright, so now I'm about to demonstrate how to use the aspirator. Now, I am right now in some purple leafy kale, which is an excellent host for some white soft-bodied insects known as aphids. Now, if you check out this leaf right there, you can see a whole bunch of aphids right there, and it'll be really hard to use your hands or a killing jar to try and do this. So, how we are going to do this is use the aspirator. So I'm going to take the mouthpiece right here, put it in my mouth, and then put the opposite end right here and suck up the aphids on this leaf. Watch. And we can see all of what we've collected in the bottom of that jar. Now we can go and analyze it, see what type of insect they are by identifying it later through our microscope, and then put it in one of those one of those vials with ethyl alcohol in it for later use. For the net technique used in entomology, you are going to approach the specimen and in one fluid motion swing your net so that you capture it and twist your wrist really quickly so that the insect gets trapped on the other side of the net. This will be useful when killing it later with your killing jar. I will now demonstrate this for this lady beetle. For the next environment that we will be checking out, I will be heading over to Bishop's Peak Natural Reserve area to be conducting a technique known as black lighting. So right now I am at the Bishop's Peak natural area in San Luis Obispo, California, and this black light technique is, as you can see here, we have a big bed sheet that is sprawled across the ground, and the concept behind it is that once it gets dark enough and it's super quiet out, there's no wind, it's perfect, it's not too cold, the insects will actually fly towards this black light and you'll be able to capture them with your bug net. So this is a really cool technique that maybe not a lot of beginners know of, so consider yourself lucky. So as you can see, we got our entire setup all ready to go, including our bug net just standing by. And as the sun is starting to go down, we are entering the perfect time of day for black lighting. It is maybe 60 degrees right now, not a gust of wind in the sky. Absolutely perfect for catching some really cool insects. This time lapse is showing the blacklight technique in action. Some other environments maybe not shown in this video are creeks, open areas, and any other ecosystem that you might think insects will be present at. Alright, so we're back and we just collected all these specimens from our blacklight adventure, and these guys have spent some time in the freezer, able to cool down, and now what we're going to do is look at these under a microscope, see if we can actually put a correct identification on the species. All right, so what I've got right here is this really cool looking moth, and I'm trying to show you without making him fall over. If you see that, he's got a lot of really cool, intricate colors. So what we're going to do is we get this guy under the microscope, and the microscope that I'm using has 3 different settings. So there's 1x, 3x, and 6x. Obviously, with the higher magnification you go, the more details you're going to be able to see. So for starting out, I always like to start out on 1x and really get a good idea of what I'm looking at. Try and find as many distinguishable characteristics as possible. And then what I like to do is bring up the magnification, move the specimen around, and try and find as much details as possible, and that will make identification a whole heck of a lot easier when we come for the later parts. All right, so now we are going to go into the identifying characteristic portion of this video, where we're going to be taking a deep dive into my insect collection to get some better clarity on what insects we have present. For the identification, I will be going over the order, the suborder of the insect, as well as the common name for clarity's sake. I will also be talking about where I found this and what the insect was doing at the time of catching. As you'll see throughout this video, there's a lot of different things going on that could really help you when finding insects for your collection. Let's get into it. When starting off our insect collection, why not go big? We're going to be starting off with Coleoptera, the order Coleoptera, which is the biggest order in the insect kingdom known currently today, with over 400,000 species. This in particular specimen that we're looking at is called a darkling beetle. Once again, its order is Coleoptera, its suborder is Polyphaga, and it's in the family Tenebronidae. Once again, Tenebronidae. Some of these names are rather difficult to say, so bear with me. This darkling beetle was found in a pile of leaf litter along the bark of a trail near Poly Canyon, and it was pretty much just basking in the sunlight trying to warm itself up, which is common for a lot of insects due to their small stature and nervous system. Moving on, we have another specimen from the order Coleoptera, and that is the western spotted cucumber beetle. Now this little guy was hanging out in some lettuce, and some more taxonomical classifications for the western spotted cucumber beetle is order Coleoptera, suborder Polyphaga, and family of Chrysomylidae. Once again, family Chrysomylidae. This western spotted cucumber beetle was seen in a lettuce field having a little snack while chewing on the outer leaves of the lettuce. This is very typical for the Coleoptera order, as they have chewing mouthparts. Our final specimen that we will be observing from the Coleoptera order is our friend the lady beetle. Yes, the beloved lady beetle is part of our collection, and what I'm going to be showing you is some different growth stages of this Coleoptera order. So the taxonomical classifications for the lady beetle is order Coleoptera, suborder Polyphaga, and family Coconellidae. Once again, that's Coconellidae. I apologize for my pronunciation. So the lady beetle starts off in a larval stage that is a beneficial insect that will eat many of the harmful insect species such as aphids and undergoes a metamorphosis in which a hardened shell comes about and essentially transitions from the pupa stage into an adult. Now, another interesting thing about the Coleoptera order that makes them unique compared to all other insect orders is this specialized forewing called elytra. So this is a hardened shell that is a modification of the forewing so that it can protect the specimen when going about its day, and it can set aside the elytra when it is ready to mobilize. This is an extremely cool order, and on to the next. And wow, would you look at that? We got ourselves a grasshopper. So the classification of the grasshopper is order Orthoptera, suborder Califera, and family Acrideidae. Once again, family Acrideidae. This grasshopper was found in a diversified field at the Cal Poly School Organic Farm. I saw it in a pasture and it was fluttering around with its long, powerful wings. And if you notice closely about this specimen, grasshoppers are known for its long, powerful legs for evading predators or bug catchers such as yourself. So they are very quick. One thing to note is the Califera suborder is not a main agricultural pest, but its counterpart, Ensiphera, the suborder Ensiphera, which is known for katydids and other such specimens, are a more of a concern to agricultural farmers and growers. Moving on to my personal favorite order, Lepidoptera. And look at these beauties. Oh my goodness. So when classifying the specimen that's seeing before you, it is in the order Lepidoptera, and funny enough, there is no suborder for these particular specimens, both of which are in the family Arabidae. Once again, that's family Arabidae. The common name of the specimen that's before you is called a salt marsh moth. One thing that I found particularly interesting about this salt marsh moth is that it's got almost like a fur coat around its collar. As you can see, a white, very hairy neck area, which almost reminds me a lot of Jon Snow from the infamous series Game of Thrones. Another characteristic about the order Lepidoptera is that all of the specimens have siphoning mouth parts. So as you see with the salt marsh moth, this long snout-like thing is its mouthpiece as a siphoning mouth part. And I collected this in my backyard doing the black light technique. So my backyard, as you'll see in a later slide, is right next to an elementary school field, so a very wide open area, and I did the exact same black light technique as seen earlier in the video. Moving on to the second specimen, this specimen was extremely hard to identify using a common name. So I was able to do some research and trying to find what common name suited best, and I found Willow Beauty, but once again, I'm not confident on that common name. It has the exact same order of Lepidoptera and family Arabidae as the salt marsh moth. And as you notice, what's really in particularly cool about the Lepidopteran order is that the wings that are present are extremely intricate in detail, color, to camouflage, to use for flight, as well as having individual scales on each one of the wings, as you can get a close-up of that picture right there. Lepidopterans start out as caterpillars, and caterpillars can be big agricultural pests, as seen with the codling moth and navel orangeworm, and some big key crops such as almonds and walnuts. This should be a relatively familiar one, as this is a common housefly. The taxonomical classification for this fly is order Diptera, suborder Cycloraphia, family Muscidae. Once again, family is Muscidae. I captured this particular specimen in my blacklighting adventure when I went to Bishop's Peak Natural Area. In regards to the order Diptera, this is the order that is classified as the true flies. So that means that they have one pair of membranous wings. And another interesting thing about the Diptera order is that the flies are holometabolous, which means that they have 4 distinct life stages, being egg, larva, pupa, and adult. We're going to now take a little blast to the past, as we're going to be now looking at the oldest order of insects currently known to man. And this order was previously called Thysanura, recently renamed to Zygomentota, and these little guys were around 40 million years ago. So that predated the dinosaurs by 100 million years. That's pretty old. This particular specimen is called a silverfish. So the taxonomy of the silverfish is order Thysanura or Zygomentota. There is no suborder, and the family is Lepsimedae. Once again, the family is Lepsimedae. This particular order of insects is unique because they do not have any wings. So this insect is wingless, and it is extremely fast, making a squiggly motion when it runs, almost looking similar to a fish. Silverfish have been known to enjoy moisture in moist areas, and the lifespan of this order can range from 3 to 6 years, and if conditions are right, some can even live up to 8 years. I found this insect in my bathroom sink. What we have looking in front of us right now are 2 damselflies. This is from the order Odonata and the suborder Zygoptera. This family name is Coenargidae. Once again, family name is Coenargidae. In the month of April, I was walking down Poly Canyon Trail, and there was a creek running down, so I went to go check it out, and I actually saw both of these specimens mating. Yes, so the blue damselfly that you see, that is the female, and the yellow or orangish one that you see is the male. These skinny, small aerial predators from the order Odonata are mainly near shallow freshwater habitats and are very, very fast, and are arguably discussed among entomologists as the fastest order of all insects. The fastest flying dragonfly ever recorded traveled at a speed of 35 miles an hour. The damselfly's taxonomical neighbor, so to speak, is the dragonfly. Unfortunately, I was unable to catch a dragonfly, even though I did find one. They are just extremely fast. The dragonfly is an insect that belongs to the suborder Ansonoptera. It is common in English jargon to be referring to these insects as bugs, but the next order that we're going to be talking about is actually the true bugs, and that is the Hemiptera. There are 3 significant suborders to this order itself, being Archanerancha, Heteroptera, and Sternarchan. This particular specimen is the common name of a water strider, and the taxonomy for it is order Hemiptera, suborder Heteroptera, family Gerridae. Once again, family is Gerridae. I found this water strider in the same creek that I found the 2 mating damselflies at Poly Canyon Trail. What makes the Heteroptera suborder unique is, unlike the Sternarchan and Archanerancha, the Heteropteran suborder has half hardened wings and the other half membranous. The other 2 suborders are fully membranous wings. The entire order Hemiptera is one of the loudest insects, mainly due to the cicadas rubbing their timbal organs on the underside of their abdomen to create a loud mating call. The adaptation seen by the water strider makes it so that its legs can use the surface tension of the standing water to create a cohesion effect, allowing them to essentially stand on water and move swiftly and skating across the water surface, predating on smaller, more helpless insects that float to the surface of the water. The next specimen that is still in the Hemiptera order is the aphid, but it has a different suborder of Sternarchancha. The family name for aphid is Aphidae. Once again, that is Aphidae. The specimens I collected were off of the purple leafy kale at the Cal Poly School farm, but the photos attached were of some cabbage that had some prolific aphid damage. Aphids especially are interesting insects, and more on a broad scale, hemipterans are hemimetabolists, meaning that they do not need to undergo a metamorphosis to form an adult between the larval and adult stages. In regards to the reproductions for aphids specifically, they are fascinating. So many aphids are parthenogenic, meaning that the females can produce unfertilized eggs, which are essentially clones of their mother. So a lot of the aphid populations are generally all females, with an exception of males to reproduce when the time comes. Lucky for me, I actually found a winged male aphid that I could add to my collection. Another rather interesting fact about aphids is that they are viviparous, meaning that they give birth to live nymphs rather than being born into eggs. Due to the fast rate of birth, it is hard to keep up in an integrated pest management program with solely insecticides. The use of biocontrol for aphids especially are helpful when using predatory ladybugs, parasitic wasps, and lacewing larvae. For this next order, we will be talking about the bee, wasp, and ant order, which is Hymenoptera. So this specimen that I have before you is a bee. So this bee is in the order Hymenoptera, it's in the suborder Opacridia, and it's in the family Apidae. Once again, that is family Apidae. Many novice entomologists have a tough time differentiating the difference between bees and wasps. And the two biggest components, I would say, to differentiate the two are their colors and their sting. So wasps have more of a triangulated head, a slender midsection, and will be able to sting their victim numerous times over. When it comes to bees, bees are a little bit less brightly colored for danger characteristics, and when a bee stings its victim, the sting is lodged into the victim's body, and when the bee starts to move, it essentially rips out the bottom part of its abdomen, killing the bee. I found this bee at the Cal Poly Organic School Farm. The other specimen that we have in the Hymenoptera order is an ant. The ant taxonomy is similar to the bee that we just discussed. So the order is Hymenoptera, the suborder is Apocridia, and the family is Formicidae. Once again, the family name is Formicidae. Much like the other specimens in the Hymenopteran order, ants are colonial, meaning that there is a hierarchy on workers, queens, and drones that is used in the survival of an ant species. Ants are extremely strong for their size, and they can lift objects up to 100 times their own mass. Some of the biology of the ant is they do not have any lungs or ears, using chemical pheromones to communicate with one another. One of the last orders that we will be talking about is the termite order of Blattidae. The suborder for termites is Isoptera, and the family is Termotidae. Once again, that is Termotidae. Much like the Hymenoptera, termites are colonial in the fact that they have hierarchical roles to develop their colony. Their colony works around 24 hours a day, every single day, so they are notorious workers. Typically, when a termite colony gets developed, it is developed in a upward fashion, and a colony can be recorded at 3 million termite specimens per colony. Termites and ants are somewhat enemies in the fact that they have the same competition of resources that they are trying to make available to them, as well as ants are predators of termites. I found these termites under some decaying bark at the Cal Poly School Farm. And the final insect that we're going to be looking at to end out our collection is actually a bonus bug in the entomology world, being the snakefly. A quick little taxonomy about the snakefly: it is in the order Rhapidoptera, in the suborder Rhaphidomorpha, and in the family Rhaphidaeidae. And I found this insect right outside my house, standing on a pile of trash. The following images seen before you are images taken from Google Maps of all of the locations that I found my insects at. The first being the Cal Poly School Farm, the second, Poly Canyon Trail, the third, West Newport Street, and the fourth, Bishop's Peak Natural Area. For the sake of brevity, I just wanted to show you my final collection of all the insects that I've collected so far. We made it to the end of the video. Congratulations, and I really hope that you learned something new about the world of entomology. Before you leave though, I just wanted to give a big shout out to two very instrumental people in the filming process of this video. The first of which is my father, Dr. Travis Hill, who's an entomologist who helped me with a lot of the information as well as some of the equipment needed for this bug collection. The second is Dr. Hedricks, who's my professor at Cal Poly San Luis Obispo, who was very instrumental in giving me a lot of good information to create some of the substance of the individual orders. Thank you very much. Now go out and collect some bugs.